Hi you guys, this is just a continuation of our biological molecules PowerPoint that we didn't get to finish, and I'm going to skip some of the activities and questions that I meant to do with you guys, so this will be a little bit shorter than if we had done it in class. So just a few kind of reminders here. We have 4 macromolecules, right? I'm just gonna zoom through the slides really quick. This was just from last week. See, we talked about bonds, talked about water, all of that stuff, talked about carbohydrates, right? Different kinds of carbohydrates, 2 types, the structure of the carbohydrates, talked about hydrocarbons for a little bit, and then we stopped at nucleic acids. So if you'd like, I'm still going to offer you the same experience as if we were in lecture, but if you want to pause right here, set yourself a timer for, I don't know, maybe 3 minutes and see how much you remember about nucleic acids. That'll help to get your brain going. So pause really quick, timer for 3 minutes, and then just unpause when you're ready. Okay, so here are some things that you should have hopefully written down about nucleic acids. There are 2 types, right? We have DNA and RNA, that's this first one right here. The nucleotides are the monomers, right? Monomers are the one piece, right? Like a Lego piece, it's one piece of a nucleic acid is a nucleotide, and hopefully you remember that they come in 3 parts. Right, they have 3 parts to them. They have the ribose sugar, they have the nitrogenous base, and they have the phosphate group. Right, and if you just look at over here on this side, this is one nucleotide. Right, here's the phosphate group. This is the sugar. And then this is the nitrogenous base. Hopefully you remember there's a few different nitrogenous bases out there, right? And we have them, it's listed right here nicely for us. Guanine, adenine, thymine, and cytosine. Those are our main 4. Hopefully you remember that nucleic acid also has a 5' to 3' orientation. So let me just check this off as we're going. If you don't quite remember what that is, it's how they're oriented. So let me erase this because it's kind of in the way. See how this one says 5', this one says 3', right? That is what that means, 5', 3'. That'll be more important once we get to replication and protein synthesis, right? So DNA can be single-stranded or double-stranded. This one, super important. This is the bond that keeps them together. This is between nucleotides. Right, so if I'm looking at, let's see if I can find it. If I'm looking at, I'm going to zoom in really quick. This line right here, where it's connecting the sugar to the phosphate of the next one, right? And then if I look right here, it's again right here. Right, connecting the carbon to the oxygen, that is your phosphodiester bond. So I'm gonna just circle those and point to it. Right, looks like that. They connect the nucleotides together. Right, connect the nucleotides together because there's another bond that is present, which is the next one, right, is the hydrogen bond. So this one is between 2 nitrogenous bases, right? So this one's between 2 N bases. So that's if I have a double helix. Hopefully you guys remember. So if here's a double helix right here, right? If I unzip that, I'm gonna unzip it right here. In between that's kind of ugly, but in between the nitrogenous bases is the hydrogen bond. So it's this blue one right there. Right, so hydrogen bond between the 2 nitrogenous bases, phosphodiester bond is between the nucleotides. Already talked about nitrogenous bases, but I hope you guys remember there are pyrimidines and purines. So if you don't quite remember what that is, I would suggest to go back and look at it, right? 2 of the 4 bases are pyrimidines and the other 2 are purines. I need you to know which one is which and what they look like, right? Is it G, A, T, C? And then what do they look like? And you can kind of have an idea also on this side, right? Just take a look at, let me change the color. Take a look at these guys, right? Look at their similarities. Again, I'm not going to ask you to draw them, but if I show you a picture like that, right, of a nucleotide, and I say, you know, what kind of nitrogenous base does it have? Is it purine or pyrimidine? You need to be able to tell me, right? Okay, so function of DNA, RNA is information storage, right? This is where we store all of our genes. This is what dictates what our cells do, all that good stuff. They do have some enzymatic activity. Ribozymes, I'll just mention that and then we'll get to what that means a little later. They can store some energy in ATP and GTP. Again, we'll kind of save that for later. Just want to put that on your radar. They can self-replicate, right? We'll talk about DNA replication a little bit later. And then DNA is stored as chromosomes. That's that kind of X shape that you guys know about with mitosis and meiosis. And then the ribosomes contain RNA, right? If you remember protein synthesis, there's a tRNA, right? mRNA, right? rRNA. Those are also nucleic acids. So this is just, again, just a little quick review about nucleic acids and what it is that they do. Remember, I said from last week, make sure you guys know the monomers, make sure you know the bonds between them, make sure you know the functions and the structure. So again, if I give you a diagram that looks like the one you see right now, whether it's a bunch of nucleotides or just one nucleotide, you should be able to recognize it. I'm not going to ask you to draw it. I just need you to be able to recognize it, right? So that is nucleic acids. Okay, here's another one that I was hoping that we could do together. But same thing, I would pause it and then maybe write down some things that you see. Right, it says that we want to brainstorm a list of concepts or terms related to protein structure or function. And we can't do a graphic organizer. You're totally welcome to do that with a friend if you'd like. But again, pause for about 3 minutes and then come back and see what you guys can come up with. Okay, so just a few things that I hope you guys wrote down about proteins, and then we'll jump right into it. But hopefully you put something about amino acids. Right, that's the monomer. Hopefully you put something about enzymes. Right, here's one that if you remember from 151, catalase was one of the ones that we used for our enzymes. Hopefully you remember what enzymes do, right? They decrease activation energy so that your reaction can happen faster, right? That's one. Let's see, what else we got? We've got, I mean, a lot of these are enzymes. Porin is a membrane channel, right? Remember, they can also be for transport, proteins, right? You can have them for transport, right? Hemoglobin, hopefully you recognize that. For my medical people up there, that is for your red blood cells have those so that they can carry around oxygen, right? Collagen is for structure, right? Insulin, hopefully you also recognize that, right? So lots of things that hopefully you remember about proteins. They do a lot of different things, right? Let me mention their bond here, which I feel like I might get ahead of myself, but that's okay. We'll do it anyway, right? They have, the bond that they have is called a peptide bond. And hopefully you remember that there's 4 levels of protein folding. If you don't remember that, I would go back to the textbook and figure out what that is. right? So let's go ahead and take a look at it. So like I said, amino acid is your monomer. This is an amino acid, right? Yes, I'm going to ask you to label the amino group and the carboxyl group. Yes, yes, yes. Right? And hopefully it's pretty easy because amino group has the N, carboxyl has the COOH. That's what they are. Right? Now you can flip that. It can be upside down. The R chain can be on the top or on the bottom. Right? So just be aware you can flip those around. The backbone of all amino acids are the same. The only one that changes is the R group. Right? And the R group can be any one of this big chart right here. Any one of these, right? These are what determine what type of amino acid you are. Now, if you're taking biochemistry, you're probably going to have to remember every single one of those, right? For our class, you don't need to. It's fine. I just need you to be aware that there are different kinds of R groups, and they do dictate the structure of your protein, right? So you can see some of them could be nonpolar, some of them are aromatic, some of them are polar, right? Some of them are positively charged, some of them negatively charged. Like I said, we don't need to memorize them. I just need you to be aware that this is where the amino acids get their properties from. Right? This is where the amino acids get their properties from. So you just have the same amino group attached to a carbon and a hydrogen attached to a carboxyl group, and then the R group can change. Right? So I can give you an amino acid that is attached to like something like this, to serine, right? But I just need you to find where the heck the other stuff is. Because if I zoom in, see how this one is kind of oriented differently? This is just an example, but I could ask you to label that. This one should be your amino group. It's kind of thick. Let me switch that out really quick. So you would say this is your amino group. This is your carboxyl, and this is your R group, something like that. Notice that they're charged because this particular type of R group that it's attached to makes it a charged particle. Or an uncharged particle, right? You might be freaking out and say, oh, well, there's a plus on the N and there's a minus on the COO. That's okay. They mean the same thing, right? Just make sure that you're aware that they can flip around. So make sure you can find the amine group, make sure you can find the carboxyl group and the R group. Okay. So we've talked about hydrolysis and dehydration reaction. It's gonna be the same for every single one. Like I said, we did it for carbohydrates. We did not do it for lipids because lipids are those weird ones where they don't really have an official monomer, so we didn't do it for that, but we're going to do it again for proteins, right? So same reaction. I can give you a example like this, a diagram like this, right? I could ask you I could ask you what type of macromolecule is involved. I'm just going to write it down. These are good questions. So keep that in mind. Try to do it for each one. So I could ask you what type of macromolecule is shown, right? And I can ask you what type of reaction is occurring, right? How do you know? That's a good one, right? So hopefully you would say, right, you'll take a look at it. Hopefully you would say you're taking water out, right? You're taking water out. In case you're like, what the heck is that? I don't remember that. I think it's, yeah, right here. It's towards the beginning, right, of our PowerPoint. If you're taking water out, that is your condensation reaction or dehydration. I'll take either one, right? If you're putting water in, that's when you are breaking it. So the diagram we were just looking at, water is being taken out. Let's see, it's this one. And you can tell because the arrow is pointing outward, right? So you took water out. So now you have built that peptide bond, which is special for amino acids, right? And proteins. So this one would be a condensation reaction. If you want to call it dehydration, I don't really care. So, right, these are the levels of organization that I was telling you guys earlier. There are 4 of them. Hopefully you remember them, but the next level that I really need you guys to know here, in case you haven't learned this before, are the bonds that are present in each one. So primary protein structure, that one's pretty easy. It's just a chain of amino acids, and they're all held together by peptide bonds. Right, and remember, because of the R groups over here, right, these R groups right here, they change and affect the conformation of the protein. So they can, go back here, they can be alpha helixes, which is kind of a swirly swirl, right? Or they can be beta pleated sheets, which is kind of like a zigzag, right? Your textbook gives you a more in-depth look of what that looks like, but just as a general sense, that's what it would look like at a glance. The bonds that are involved here a lot are the hydrogen bonds. And again, that happens a lot because of the R groups that are present on there, right? Next one is your tertiary protein structure. So if you have a combination of the alpha helix and the beta pleated sheet, they become more globular. Right, now this one's going to be a little bit different because you might not have heard of these bonds before. We talked about it a little bit at the beginning, but for tertiary, the ones that work on them a lot are going to be the electrostatic interactions, right? So let's see right here. So chain interactions, you have hydrogen bonds still, right? But you could also have disulfide bridges, right? Disulfide bridges, and really also van der Waals. If you don't remember what those are, I would go back to your textbook and take a look at them. But pretty much what they are is they interact with specific charges, for example, right? The disulfide bridges, just as it sounds, it's 2 sulfides together and they make a bridge interacting with each other, right? And then when you have 2 or more of those tertiary protein structure, then you have your quaternary. Usually that one is characterized by different colors. That's usually what that looks like, right? Different colored globs is going to be your quaternary structure. Now for quaternary, that one doesn't have a predominant bond. You can still call everything in there, right? You can still consider all of those in there to be in your quaternary, right? The most common one will probably be your disulfide bridges though. Right, so then here's another activity. So same thing, I would pause it, wait 3 minutes, and try to label it. So we just talked about what they look like, plus the bonds that are associated with them. So I would suggest you do the same thing. So pause for 3 minutes, label it, what kind of level of structure it is, and then label what kind of bonds are present there, right? And then just unpause when you're ready. Okie dokie, here we go. So letter A, that looks like a alpha helix, so hopefully you put secondary, right? Letter B looks just like a straight line. It doesn't have that swirl or that zigzag, so this would be our primary. And we'll go back and do the bonds in just a little bit. Letter C, it's behind something, and we'll try our best to do that, but it still looks like a strand to me, so that'll be primary. Letter D has a little zigzag thing going on, right? Little zigzag thing going on is our beta pleated sheet, so that would be our secondary. Letter F, same thing. It still kind of look like a string with no zigzags or swirls, so this is our primary. And then this is the weird one, right? This is the one where it kind of looks like it's a zigzag, but it's a different color. The clue here that I put for you is it says it's a type of covalent bond, right? So between these two, so if we just look at the list here, right, between these two, it's going to be some kind of bond that interacts between the secondary. So spoiler alert, it's going to be a tertiary, right, because it's interacting with that. So this would be a tertiary, and then the whole thing is a tertiary, right, because it's just one color. If there was another one in here that was like a different color, then it would be quaternary. But because they're all green, it's going to be tertiary. So let me go back and do the bonds though, and I'll explain what that little lightning bolt is about. So primary ones, hopefully you put peptide, 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 right? For the secondary one, hopefully you put H bond, right? And that's the predominant one, right? I'm not going to try to trick you and ask specifically between specific proteins what they are, but just in general, they're that. And then for tertiary, this kind of lightning bolt thing right here, that's your disulfide. And I know it's kind of hard to see because there's no S's there, but that little kind of lightning bolt thing going on there is a type of a covalent bond. Right, so a disulfide bridge is an example of a covalent bond, right? And then for the whole thing, I mean, you know, you can go back to this list and just mention all of it, right? Hydrogen bonds, disulfide, and van der Waals for the entire thing, right? So also a really good test question. I can give you the same exact protein. I can give you something slightly different, ask you to label the foldings that are present and the bonds that are involved in those levels, right? Okay, so nowadays, we can pretty much characterize a lot of the proteins we know of using like computer technology. But before that, when we were still trying to explore proteins and figure out what they were, these are the two techniques that were used. There's X-ray crystallography and NMR. Right? And NMR, I'm pretty sure you learned that in organic chemistry to read an NMR sheet and stuff, but that's what they use to figure out protein conformation. Right? Another word for conformation is shape, just in case, so you don't get confused. This is how they figure out the shape of a protein, right? And I guess I should say you are welcome to use X-ray crystallography or NMR as your project if you would like. I don't remember if I put that on the list. Okay, so here's an example of a quaternary structure, right? So this is an example of a quaternary structure. And how do I know that? It's because it's a bunch of tertiaries that are different colors, right? That are different colors. Now, the significance of this slide really goes back to the shape of it. So we have hemoglobin. If you remember what hemoglobin is for, it is that compound in your red blood cells that help you carry oxygen around, right? So normal hemoglobin is the top part, and then the bottom part is the sickle cell hemoglobin. I'll talk about that on the next slide. But again, for proteins, it's shape equals function. If you need a new tattoo, that is your new tattoo. If you love proteins as much as I do, right? Shape equals function. If even the tiniest thing changes about a protein, is not going to work anymore, right? It's not going to work anymore. And that's the problem with this sickle cell hemoglobin, is it doesn't look anything like the other one, right? The structure looks different, even though the same proteins might be involved. There's just something different about it, right? And just look very closely. So for example, here's like the top part is yellow, the bottom part is blue, whereas here it looks like it kind of split itself almost into two. And then there's these red dots on the top one, but purple and green on the bottom. So that's kind of weird, right? So I can ask you a question like that and ask you, describe what's happening in the picture, right? And then you can just say that the protein shape affects the function, right? So because the hemoglobin, the second one, sickle cell hemoglobin, no longer looks like the original one, it doesn't do the same job. So where does this stem from? It stems really from the DNA of it, right? This is a mutation. So if we take a look at this box right here, right, the first line of amino acids is the normal hemoglobin, and the bottom is the mutated one, right? So everything is the same except when we get to number 6, right? Everything is the same amino acid wise until we get to 6, and that one drastic change, right? And it's just one. This is just one amino acid, just one, right? That drastic change, changed the shape of the protein, which means that it changed the function, right? So how can a single amino acid change or alter the shape of a protein? It can do it quite a bit, right? Now, there are some mutations where it doesn't do anything. Like, yes, you change the DNA, but the protein is unaffected. That's fine. But this one is just such a drastic change. So in case you're not aware of what sickle cell anemia is, our red blood cells are usually kind of like a donut shape like that. Actually, let me put it right next to it. Right, it's kind of a donut shape like that, our red blood cells, and it has hemoglobin inside. With sickle cell hemoglobin, it actually also even alters the shape of the red blood cell itself, right? It looks like a sickle, like the ones that you would use for farming, right? Which then decreases the level of oxygen that you're able to transport around your body. Okay, so here is a bunch of proteins, right? And hopefully you can tell me, hey, these are tertiaries because they're all just one color. I can see squiggles and zigzags, so these are all tertiaries. The main idea here, right, is that even though you can have different types of sequences, they can still fold to be the same shape. Right? So here on the first column, the green ones, we have actin, MREB, and PAR-M, and they have about the same shape, right? You might not really see that because you might be nitpicking and say, oh, well, no, actin looks a little bit different, and so on and so forth, but it's the main shape of it. I personally, I prefer the string one that looks like this, so I can see the alpha helices and the beta pleated sheets. Some computers can do the bubble one with the globular proteins, right? But the main idea here is proteins with similar shape will have similar functions, right? We know what actin does. Hopefully you guys know actin is a structural protein. So anything that has the same shape as actin, then I can assume that MREB and PAR-M also probably is a structural protein of some sort, right? I don't really need to know that exactly, but that could be a question, right? If I gave you a shape that is like a oval like this, right? And I have 3 different proteins that are about the same oval shape. One of them might be a little bit longer than the other. One might be a tad bit smaller than the other. But then you can assume that all of those mean the same thing, right? And this is something from the paper that hopefully you caught, right? All of those circular little things from one of the figures, they're all circles, right? And even though you don't really know what they all look like circles, which means that they probably have the same function, right? And this is just us thinking about that or inferring that, right? And as a scientist, if that was you doing that experiment on that paper, as soon as you think that's what they do, then you go ahead and test that, right? But again, shape equals function. So if they have the same shape, they probably have the same function, right? Let's see. So subsections of proteins or domains, that's what they're called, can fold into specific conformations that are associated with specific functions, giving a protein the capability of performing complex tasks. So you guys will see the word domain and motifs again, right? That's just a protein word that we use. But each one of them can do different things. So like this whole thing right here, right? That's one protein. That is one protein. This would be a quaternary protein because there's 3 colors. What this diagram is telling you is that if we break the protein apart, right, you can see that they all fold differently, which means they do different things. And this is going to be, again, related to the paper. And I guess this might be the downfall of us not getting to this protein conformation lecture, is you might have struggled a little bit on that paper. So I apologize for that. But if we unfold the protein a little bit and break it apart, we can see, like I said, that they can do different things. And that's the cool thing about proteins, right? Is they do a lot of cool things depending on how they are put together. Okay, this one, last quick activity for you that I want you to try. Same thing, pause it, give yourself 3 minutes to write a short figure legend for what's going on in this picture, right? Again, this is a 300 level class, so I need you to be able to take a look at figures and write something about it based on what we're talking about. So pause, give yourself 3 minutes, and then I'll show you the answer. Okay, so here's the answer right here. Hopefully you got similar, right, sentiments to what I'm saying. The main idea is that proteins can change. That's it. Proteins can change shape. If there's something that binds to it, which in this case is CAMP, hopefully you remember that. That is a second messenger, right? Anything that binds to a protein, if something binds to it, it will change, right? Whether that's the substrate, whether it's allosterically inhibited, whether it's competitively inhibited, every time something binds to it, it changes the shape of it. So what this picture is telling us, right, is that this protein, the red part is the DNA binding domain, and the bottom part is the CAMP binding domain. So what it's saying is that when CAMP binds, which is going to be these purple guys right here, right? When CAMP binds, it changes the shape of this protein, which then allows it to bind to the DNA. That's what that means. So without the CAMP, so I could ask you a question. So what would happen if there's no CAMP? Right? No CAMP. So what will happen? DNA cannot bind. Right? And then whatever reaction this is doing cannot happen anymore. Right? So this is very, very important. So important that I took it from my undergrad and kept it here, is Dr. Kasim. If you know Dr. Kasim, she is so, so awesome. She was my PI and the one that really helped me fall in love with with cell biology. She works at a different department now. But Dr. Kasim's rule for protein conformation is just what I said. Whenever something binds to a protein, right, or releases from a protein, then the shape will change. And if the shape changes, the function will change. Right? So always, 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 shape equals function. Right? Shape equals function. There is a special word that I'm just going to bring to your attention. I hope you guys remember what it is, but it's the word denature. Right? If you don't remember what that is, you might want to look it up, because as we continue to talk about proteins and eventually we'll talk about enzymes, I'm going to bring up that word again. So I need you to know what it is. But that's the end of this lecture, and then I'll see you guys in the next one.